Chapter 12, Characterizing and Classifying Eukaryotes. In this chapter, we're going to cover the following characteristics. General characteristics of eukaryotes. Then we're going to talk about the protozoa, fungi, algae, and then we will finish up with other eukaryotes of microbiological interest, basically the worms and the vectors. Now, because this overview is so short, we're going to launch right into general characteristics of eukaryotes. So, general characteristics of eukaryotes, Chapter 12. The learning objectives for this topic are: state 4 reasons why eukaryotic reproduction is more complex than prokaryotic reproduction. Describe the phases of mitosis, mentioning chromosomes, chromatids, centromeres, and spindle. Contrast meiosis with mitosis, mentioning homologous chromosomes, tetrads, and crossing over. Distinguish among nuclear division, cytokinesis, and schizogony. Briefly describe the major groups of eukaryotes as they were classified in the late 18th century and as they were classified in the late 20th century. And list some of the problems involved in the classification of protists in particular. In this topic, we're going to talk about reproduction, nuclear division, cell division, schizogony, and classification. Now, under nuclear division, there are 2 types for eukaryotes. The first one is mitosis, and that involves division of a parent cell into 2 cells that have exactly the same genetic composition as the parent cell. Now, the other type of nuclear division is meiosis, in which we have a parent cell, but at the end we have 4 daughter cells that are haploid and have a different genetic composition than the parent cell has. Now, we will have covered this in detail in Biology 201. I'm not going to cover it in detail here, but if you want to review, I'm going to make my lectures for Biology 201 that cover this topic available to you, and I'll also have other professional videos available for you so that you can meet the learning objectives covered under the topic of nuclear division. On to cell division. You've probably covered cytokinesis or cell division as happening at the same time or immediately after telophase of mitosis or meiosis. But in this class, we're going to be discussing some of the variations on the theme, if you want to call it that. So, to remind you, in animals, after telophase or sometimes during telophase, we get a cleavage furrow. There are actin filaments that form kind of a belt underneath the cell membrane of an animal cell, and it tightens and tightens and tightens until the cell cytoplasm is cleaved. Now, plants have a cell wall, so they can't do this. So after mitosis or meiosis, a cell plate forms, and these are vesicles that contain enzymes for forming a new cell wall, and the cytoplasm is divided. Now, fungi, especially yeast, can do cytokinesis or division of the cytoplasm asymmetrically, so that we get a little baby cell budding off, and then the cells are divided, and then the little baby cell grows up to be a full-size cell. Now, while we're on the topic of fungi, some molds, some multicellular fungi, do not undergo cytokinesis. They undergo mitosis, and the cell enlarges, but the cytoplasm is never divided, or not divided completely. Now, you're probably wondering, how in the world does it handle getting that big, because there's a problem with diffusion of nutrients into the cell and waste out of the cell? Well, fungi manage this by being filamentous. They're long, they're thin, they have a large surface area for their volume, and they have what's called cytoplasmic streaming, where nutrients are transported from one end of the gigantic cell to the other end. Now for the last thing, schizogony. Some cells, especially when we talk about malaria, undergo multiple phases of mitosis. like the fungi do. But instead of being filamentous, they remain circular, and the cytoplasm does not increase in size. Then they will undergo multiple rounds of cytokinesis so that you get all of these very small cells, each containing a nucleus. And we'll talk more about those when we talk about some of the protists that undergo this type of cytokinesis or cell division. Now on to classification of eukaryotes. And as you can see from my quote up here, this is a quote from me, it's a mess. So let's look at the history and why I consider eukaryotic classification to be a mess. So Linnaeus in the late 18th century came up with basically two kingdoms, Plantae and Animalae. Now later on, when we were trying to include fungi and different things, it didn't quite work. So Whitaker came up with fungi, Protista, to handle single-celled organisms that originally had been in Animalalia. And then we have Plantae for the plants and Animalalia for the animals. Well, just like what happened with the bacteria, with discovering that archaea weren't terribly related to other prokaryotes, when we've done genetic research, and this would be in the 18S rRNA, we found out that a lot of the protozoa, or protista, weren't terribly related to each other. Some of them are going to be put in with Plantae. Some of them are more closely related to animals and fungi. Some of them may end up with their own kingdoms. We're still working it out at this point. That's why I say it's a mess. Give us another 10-20 years. We'll have things decided. We'll have set new kingdoms and different classifications, and it'll all be worked out. But right now, it's in transition. Part of the reason that we are in transition is for the same reason that we had to transition from talking about just bacteria and splitting that group into bacteria and archaea. With the protista, anything that wasn't obviously a fungi, a plant, or an animal, and it was eukaryotic, mostly, or single-celled most of the time, where most of the species are single-celled, got tossed into protista because they were all single-celled and they all pretty much look alike. But once we started doing genetic research, we find out that they're really very different. Just like we found out that archaea are really very different from bacteria once we knew to look for the differences. And the same thing is happening with the protista. So that's one of the problems with how classification arises. Originally, when we were setting up protista, we broke them into how they moved, by flagella versus cilia versus amoeboid movement, which is all fine and good when that's all you've got, but we've got to rearrange things once we come up with a better system. Now, because we're in transition, depending upon which textbook you look at, you may see differences in how the protista are presented. I wouldn't let it bother you. Just go ahead and learn the material as best you can and trust that we're going to get things solidified eventually. Well, that's it for this topic. Here is a summary of the differences between the two types of nuclear division, mitosis and meiosis. And here are the reminders of what you need to learn from this topic. Remember to write down any questions that you may have so that you can ask them in class.